Welcome to The Edge, a program where we spotlight Purdue University faculty, scholars, and innovators, highlighting their careers and scientific achievements and their vision for the future of their scientific field. My name is Tommy Source. I'm Assistant Director of the Purdue Institute of Inflammation, Immunology and Infectious Disease. And I welcome you again to another feature professor who works on microbiome sciences, Dr. Tzu-Wen L, Assistant Professor of the Department of Nutrition Science at Purdue University. Welcome, Tzu-Wen. Thank you so much for talking to us.
Yeah, thank you so much for having me.
I have been looking forward to this interview for a long time because I admire your work. I think you are doing really exciting work that helps understand the differences of gender and hormones and microbiome and nutrition and health. And it's such a hot topic. And people are so interested in this field. And I can't wait to hear a little bit about you, what you're doing with this work, how you're moving the edge of knowledge and the edge of this technology forward. And, I really thank you so much for coming on and talking to us.
Yeah, for sure. I'm excited to share some more about what I know and what I don't know today.
Absolutely. Well, we're excited to hear about it. Maybe you can take us back and tell us a little bit about your education, your background, where's your training from and all of that.
Yeah, sure. Let's see. I guess we can start from undergrad. So I grew up in Taiwan. My dad is actually a pharmacist. And we own a pharmacy. That's how, where I grew up. So for a long time, I thought that pharmacology is where I wanted to be. I don't know if it's the same for everyone, but it seems pretty common that what your parents do is what inspires you. Up until one day, I was one of those kids that always ask questions, ask why. And I asked him a question about why do we use this drug versus that? And he started drawing the structure of the drug and told me, this one has this benzoic ring and that's why it does this. And I just thought to myself, I don't want to memorize all of those. I want something that's a little more applied, like in my head at the time. So I started thinking what other things I wanted to do. And the culture that I grew up is we value food and nutrition a lot. And that is something that we always wanted to eat well, have good nutrition. So the preventative medicine part through food is big. So that started to got me interested. And I thought to myself that, even if I don't ended up doing anything with nutrition or dietetics, it's something that would benefit me or benefit my family. So that's ended up being the path that I head towards, at least. I went to medical university actually with a dietetics track, a nutrition science, human health dietetic track, ended up getting my license in Taiwan as a dietitian. Throughout along the way, research always interests me. I was a part of a lab for most part of my undergrad and thought that that's something I wanted to do, but then all the PhD students at the time, were spending, 8 years, 7 years, 6 years, long period of time in getting their education. So I thought I got to make sure that this is what I want to do before I jump in. So I worked a couple of years as research assistant and also part-time dietitians. I was really trying to convince myself to stay in clinical work. It'd be research. It sounded like a long path, and it could be lonely at times. So I was trying to convince myself to stay in the clinical world, but there's something about research that keep drawing me back. So anyhow, I ended up going to grad school. I got my master's degree from the University of Missouri in Columbia, where I work with a couple of people who I admire and really enjoyed my time there. And then in nutrition science as well. So I was doing a lot more lipid metabolism in liver and a little bit of adipose tissue, looking at tissue crosstalk of lipid metabolism. And then I thought to myself that I've been doing lipid metabolism for a while when I was an undergrad and then research assistant. And gastrointestinal tract just keeps coming back, because when we think about nutrition, that's the first exposure, right? Our gut sees what we eat first. So then for my PhD work, I really wanted to get into intestine. So I ended up doing my PhD at the University of Illinois with Kelly Swanson, learning the gut microbiome and how nutrition can impact us, metabolism, physiology, but also through the microbes that live in our gut, and how that can indirectly or potentially directly then impact us. So I guess that's sort of my path. Oh, and then with most of the nutritional studies, microbiome is seen more as a secondary analysis, since we really wanted to see outcome from weight gains for metabolic disease or insulin, those other type of markers. Microbiome typically is seen as a secondary outcome. I wanted to learn ways that we can have a more a clearer understanding of how the microbes can directly impact us. So for my postdoc, I joined a lab that at University of Wisconsin with Federico Ray to learn how to work with gnotobiotic mouse model or germ-free mouse model, where we can then colonize these mice with any type of microbial community that we're interested in. So that's sort of the path that brought me here.
That's awesome. That's such a, it's incredible. Again, I see this time and time again, how an initial inspiration with so much focus, like you carved a path, but it was always in this nutrition path and very focused on that application. And it's so admirable to see that directedness. Tell us a little bit about this postdoc though, because this seems really interesting. Please explain for those that don't understand or have never heard of this gnotobiotic mouse. What does it mean? Like, how do you work with them? How delicate is this? And how much information can we get from these types of models?
Yeah, that's a great question. So gnotobiotic means known life, means that we know what we are, or what we know what's present within that ecosystem. So it's a tool that we can use to then understand what a given ecosystem can do, what type of metabolites they can excrete, what type of gene function that they're capable of doing for us or for them, and then indirectly impacting us. So the model that we are using right now in the lab is a rodent-based model. So that these are germ-free mice. Because bacteria, microorganisms are everywhere around us. So it is quite difficult and delicate in terms of being able to make sure that these animals are clean, are truly germ-free. This is not the same as sterile in terms of a surgery, right? This is truly sterile. We cannot tolerate any type of organisms to be present. Wow. So we have to house and raise these germ-free mice that have been re-derived germ-free by people, a long time ago, that they live in this plastic bubble, vinyl bubble. Everything that goes in needs to be autoclave or completely sterile. Everything go out. We need to make sure that the port access won't potentially introduce bacteria coming in. So it takes a lot of training, dedicated people who knows how to make sure that contamination does not happen. So we have these germ-free colony, these mice are born and raised without any microorganisms. At the time when we wanted to do experiments, so say that we have a community, it could be from anything. It could be from human, it could be from another mouse with certain diseases that we're interested in. It could be from another species. For example, I'm doing some work with Tim Johnson at Animal Science right now. We're about to transplant microbiome from pigs into mice as a model, because having germ-free pigs is another huge undertaking. So there are different things that we can do in terms of that initial community, where we extract that community from, in terms of the host that's associated with them. We can also do what we call a synthetic community, where we culture bacteria individually or together, depending on the study design. But these are known bacteria that we purchase individually and pool into communities and colonize them with, it could be differ by one strain, it could be differ by one function that we're interested in, and design community that way for more of a proof of concept type of experiment. We can, of course, also do mono association, just one strain of bacteria and colonizing, and compare that with germ-free. People do that as well. So
you're really engineering the microbial ecology of that animal by inoculating it, introducing these bacteria into them, and then studying how, or this association with other species or other communities. So interesting. This is again, like, I don't know how many people know that this really exists, because, you know, like, you talk about how difficult it is to maintain an environment without any bacteria, none, no fungi, no bacteria, nothing, no spores, nothing in the air. No viruses. Wow, that must be unbelievable. And the training. But, you know, Tzu-Wen, I think for a lot of people, they would say, well, you know, our parents said it's okay to have a little cold here and there so that you boost your immune system. Are these mice sick, or what's their immune system look like if there is no insult from bacteria or virus or fungi that are invading them constantly?
Yeah, that's a great question as well. And if you think about it, it's not even just the insults, so to speak, not just pathogenic, right? There's a lot of immune learning that our body learns after we're colonized with what we call commensal bacteria that were just there. They're not causing any issue for us. Our gut is colonized with these commensal bacteria soon after birth. So there's a lot of immune learning between the microbes that we acquire after birth and throughout life. So you're absolutely right. These mice, germ-free mice, are not normal in a way that we think about animal, think about human. This is a debate, but in general, more sterile in utero. There's some data showing that there could be some microbial presence when we're in utero, but the biggest load of microbes that we're encounter is definitely after birth, either from the birth canal or from the skin of the person who are holding you, if it's through C-section. So germ-free mice is definitely in a very artificial environment that we are maintaining them, that they don't get that exposure that they would have otherwise exposed. And their immune system is definitely not as strong compared to regular mice. We have, in my experience, we try not to colonize these mice too young. We have higher mortality when we colonize them right after weaning. For reasons that I still I'm not 100% sure, but my hypothesis and thoughts would be that there's a huge amount of microbial load that these mice then or their immune systems have not been trained gradually throughout that developmental process. And then all of a sudden we give them a community from an adult animal that has all sorts of things. So the mortality in our hands are typically a little higher at that early of a phase. Physiologically and anatomically, they're also different. Their gut is a lot bigger and looser. The tissue, when you look at them compared to a regular animal, they're very stretched out, very thinner. It feels like their tight junction that holds the cells together are not as intact. Their cecum, mice are cecal fermenters. Their cecums are huge. When you open, dissect these mice that are germ-free, that's really the first thing you can see. They're huge compared to regular animal. And once we colonize them, you can start to see the size of the cecum shrinking. The tissue seems to normalize along the way. So it's very interesting how the microbes in our gut can then impact the physiological and anatomic differences. And interesting you asked that. It seems to potentially impact how we acquire nutrient as well. And this is one of those unexpected finding that we found that I, again, still don't know how to explain it. I have some hypothesis, but when I was doing my postdoc, my mentor had a thought, and we started talking about it, is that we typically say that microbes are important in terms of when we consume dietary fiber, but we don't really know that for sure. So I designed different types of diets and started to feed germ-free mice with these diet that contain different types of fermentable fibers, to see that if it's true that without microorganisms, we're not able to obtain any benefits from consumption of dietary fiber, right? We can't, mammals or humans do not have the enzyme to break down, mainly human, cows can, but we don't. Our mammalian cells, at least, do not have the enzyme to break down some of these linkages. So we're really relying on the microbes in our gut to do that. And so when you don't have it, do you still gain some of these benefits? Certain dietary fibers that I tested, all the groups of that mice that are germ-free died. So it seems like there could be some osmolarity issues, that you really need those microbes in our gut to maintain that normal physiological functions, and to the point that dietary components could be lethal. It's very interesting, and we don't really know a whole lot about.
So fascinating.
Yeah.
Oh my gosh. So fast. There was so much that you said there that just like blew my mind. That was so great. Wow. What an incredible set of information that you're providing us here, because it just like that the microbes can have such a huge anatomical effect, and that they would have obviously such a big immune effect too, and their sensitivity of these mice. But now you're telling me also this big impact on the way that we digest, the way that we can or can't digest, and potential impact of that. So my gosh, where should we go? Because I have a lot of questions and a lot of ways that we could ask this. So tell us a little bit. Okay, so you also said not only does this have an impact on our immune system, but that there is a certain load of bacteria also, that if we take it from an adult and we try to introduce it into a juvenile that has or a gnotobiotic mouse that doesn't have that type of profile, that we could shock them and it could create problems and disease potential, right? So this connection between microbiome and chronic diseases, and I mean, you are what you eat, Tzu-Wen. Is this what we're like? But but so then like the colonies of microbes that are in our gut that are commensal, they are really helping us. They're not really, it's commensal. We don't see that they're not, we see that they're not pathogenic, but they're really symbiotic in that sense that we need them to help us digest and survive. And if we take what your observation is true in the mice, right?
Yeah, that's what it seems like. If we live germ-free, that would potentially happen in terms of the specific types of fiber that we saw. So yeah, your conclusion might actually be true, which we will never know, right? We will never be able to or want to really
Right.
into humans to be able to test that. But yeah, the thought of colonizing a very young animal with an adult profile of microbiome, and that's what's causing it to be more lethal, it was my thought and hypothesis. Never tested it, but thinking about when we're born, the microbiome, if you track the profile from a baby and then all the way to mature, juvenile and then adults, the profile keeps changing. We are at a young age, it's mostly Lactobacillus bifidobacterium, a lot more simplified profile, and also because the diets, right? When we're that young, we're consuming mostly milk. So the dietary substrates that can get into the gut has a lot of impact on what kind of bacteria can survive there as well. And as we starting to introduce solid foods and more complex carbohydrate, lipids, proteins, and that when we would then starting to be able to promote and support different growth of various bacteria. And of course, we're self-inoculating as well from food, from other environmental factors that can then introduce different type of bacteria. So it's a much more gradual process in terms of load and diversity of the microbes that we can harvest in our gut or harbor in our gut versus what we do again in the lab more artificially, that we're exposing them to something it's very diverse, right? So that was my thought.
Yeah, no,
Yeah.
what it seems like it has a lot of good rational thinking in there. And maybe at some point in time, I know this is probably goes beyond of where we want to take this conversation, but as we start to look at space travel, and we're looking here at Purdue for the Space Force and trying to look at what it would be to colonize the moon and so on, maybe some of these questions do become relevant, because what we don't want to inoculate the moon or Mars or anything outside of our Earth with the bacteria that we're carrying. And I know NASA is very concerned about that. But regardless, I what an interesting concept to think about this connection. And I know you did incredible early work to talk about this connection also with fats and obesity and lipids. Tell us a little bit, if you can take us back to some of that work. And I know you continue to be doing some of that work now as well.
Yeah. So I've always been very interested in metabolic diseases or metabolic dysfunctions that are associated with obesity. So the earlier work that I mentioned earlier with the lipid metabolism, we're interested in how dietary fat gets metabolized and what type of fatty acids we then able to absorb, getting into the systemic circulation, how that impacts the profile that our tissue acquires that's in our systemic circulation, which has different species of fatty acids have different impact on metabolic processes. So those are some of the things that earlier on that I'm very interested in. I'm still very interested in that.
Yeah.
Fat is something that's can be a little tricky to study with the microbes. So that's a link that I'm really looking forward to connect. Haven't figured out exactly how I wanted to approach that part yet. But when I move towards learning the microbiome part of the work, mainly we're looking at metabolic outcomes. So we're still incorporating a diet that are higher in fat, higher in fat that's more common in human diets, proportionally, and the type of fatty acids that are included in those diets, but more so thinking about the substrates that our microbes can encounter more so. So things that we cannot digest, or things that we're not able to absorb very well, such as some of the soy isoflavones that we're continuing to work on. Our body, some certain types, we don't absorb those things as well. So then that gets carried all the way into the large bowel, into the colon, so that the microbes now get to see them. So they can acquire certain parts to generate energy for them. And this is the same with the sex steroids that we're interested in as well. The conjugation part of those compounds can oftentimes be cleaved by these microbes, and then used to generate energy. And then what's left, then we might be able to reabsorb and impact our cells. They can also do conversion of these compounds to make it more biologically active for us, so that impact bioavailabilities. So those are the things become something that we think about a little more, just purely from the part that's the substrates that can go all the way and the microbes can see. Fatty acids, of course, some of them can travel, but a lot of the time, 95% of the dietary fat that we consume are absorbed. So the impact potentially is more indirect from bile acids, from metabolic, right? We're absorbing that and storing those in adipose tissue and potentially impact inflammation and cytokines and all those things. So a little different thought process.
And Tzu-Wen, do we actually engineer our microbiome, our gut microbiome, with what we eat? So it's not like you can get skinny by obtaining the microbiome from a skinny person and replacing my, fattier microbiome. Is that even possible? Or do you really have to train it? Your microbiome needs to be trained by the consumption of particular foods that already have this inoculum of bacteria that pounce on those foods and can digest those foods, right? So we're bringing that in. Is that the way that this is going to go? And because I couldn't see us doing microbiome replacements from skinny people into fatter people, and then that will solve their problem.
Yeah, there's a few different things to think about from the question you ask. It has been shown that if we transfer an obese either person or mouse microbiota into germ-free mice, that can make those mice gain more weight compared to if they receive a skinnier person's microbiome. So that's a possibility, but I agree with you that we don't really want to go around and inoculate people with microbiome from skinnier person. If you wanted to, if weight is a concern, one is, depending on what you have, your existing commensal microbe, your community, that may or may not work. If there's not a niche of, depending on the hypothesis, if we think it's a particular type of microbes that's doing that for us, you may or may not have the niche in your existing community for those microbes to colonize. If it's a much more complex ecosystem interaction, microbe interactions, that is affecting nutrient processing and metabolism of these microbes that ended up generating different type of metabolites for us, that would be much more complex than just replacing a person's microbes, right? And also need to keep in mind that we acquire microbes all very differently, right? From our mom, from the hospital staff at birth, and then throughout life, every exposure, where you grow up, what kind of water you drink, all of that affects things other than the diet. But thinking about an adult, since what happened already happened, there's a lot of things we couldn't change in the past, right? So what we already have in our gut, can we manipulate that with food? We certainly can to a certain degree. There seems to still be a very individualized microbiome. When we look at profile of a person, even if we do a lot of the dietary intervention, they tend to still look more close to themselves. You know, if we introduce different type of dietary substrate and do repeated collections of a person, we're still seeing a lot of interpersonal variations of the microbiome. So there still seem to be a signature, not only what you expose environmentally growing up, but also from your genetic components that potentially can affect what microbes would preferentially colonize and thrive in that community. So I think it's at least the way that I think about this question, it's both and. We definitely need to think about the substrates. I think about dietary substrates a lot more, but of course, pharmacological, sex hormones, something that we're interested in as well, and that seems to affect what mice are able to colonize, whether you're male or female. So things like that definitely has an impact on the community that a person or an animal is able to support. So thinking about it more from two angles, probiotic or FMT or fecal microbial transplant that able to replace or restructure that community to our advantage, but also continually having support from diet, from these other modifiable factors, how I think about it, would be important. This is not something that we can just eat healthy one day and then go back to eating unhealthy and thinking that we can continuously getting that benefits, right? So both things are important, I think.
Yeah, and I think you paint a picture that we need to think about this in a holistic, more broader way than just punctuated, acute, set of circumstances. The connection with disease is also so interesting, and some of the bowel diseases or digestive diseases or related diseases that can arise with that. I know you mentioned this word probiotic, and I know you can probably be one of the best people to explain to us what is the difference between a prebiotic and a probiotic.
Yeah. So prebiotics are dietary constituents. So some dietary fibers, so these are carbohydrates that are not digestible by us, by the mammalian cells. They're able to be, some of the components are able to be digested by the microbes that we have in our gut. Certain types in particular of the dietary fibers can be classified as prebiotics when the definition of prebiotic keeps changing. But in general, we need to first isolate those compounds and be able to show that those compounds in particular can promote the growth of good bacteria. In general, what we think about that right now is Bifidobacterium, lactobacillus type of bacteria. So that needs to, we need to be able to show that for it to regulatory be able to be classified as prebiotic.
Got it.
Probiotic, on the other hand, is the bacteria. So if one does not have certain type of bacteria in their gut, and we introduce this probiotic, and we don't have enough or the right type of prebiotics or dietary substrate to promote the growth of that probiotics, that wouldn't work as efficiently as if we have both things, right? We're introducing the bacteria and giving them the food that they like to eat to support them. So when you give a person both pre and probiotic, that's when we call symbiotic.
Got it. Yeah. Got it. Oh, it's so interesting. And so me being, my background being in plant biology, you talk about, oh, soybean isoflavones, and I'm like, oh my gosh, okay. Well, like, are there bacteria that are in our gut that are benefiting from this? And what is the impact of those in our own hormones and the connection with the microbiome in our guts and hormones? What can you take us a little bit and uncover that for us if you can?
Yeah, that's something that we're very interested in trying to learn as well. So one of the soy isoflavone called daisine, it's actually something that the microbes is able to convert into a compound called S-equal. S-equal is a compound that structurally look very similar to estradiol or estrogen.
Estrogen.
Yeah, and equal is able to preferentially, they can bind to both genomic base receptor, estrogen receptor alpha and beta, but seems to preferentially bind to the beta form more so. So it has been hypothesized that a person, if you have a way to make that conversion, so convert daisine into equal, at least epidemiological study have suggested that that seems to can benefit a person more so from consumption of soy. So that interpersonal variation comes out from studies like this, and also larger clinical trial that when we feed people the same exact diet, not everyone is going to benefit from them. Of course, there are other components, right? Genetic, epigenetic, those things can contribute, but certainly it seems like microbiome can be a determining factor as well. In terms of soy that we've talked about, the ability for a person to make equal. So if you have equal producing bacteria in your gut or not, it seems to determine whether we can acquire benefits. Especially a lot of work have been done in postmenopausal women. When we lose that endogenous synthesis of estrogen, or majority of them in that postmenopausal phase, increased consumption of soy seems to be able to alleviate a lot of the side effect, a lot of symptoms that happen associated with that menopause transition, likely because of that structural similarity and able to bind to these receptors to alleviate that sudden change of hormonal profile. But interestingly, even more interesting, I think, is that only globally, about 30 to 50% of the people are able to make equal. So only about half of us is equal producers. And it seems to have geographical differences as well. In Western country, in North America, only about 20-30% of people are able to make equal from soy isoflavone. In countries that habitually seem to consume more soy, in Asia, more so like 50-60% of the people are able to make that compound, a conversion. So we're interested to see if this is habitual diet related. Are we able to, by introducing more of soy in our diet here, able to make more people to become equal producers? I think it's probably need to be that double-edged approach, that we need to have the dietary substrate to support that, but also the equal producing bacteria we probably need to introduce, because there could be intergenerational loss, that your grandmother's not really eating soy, your mom is not eating soy, and then throughout the years we just lose those bacteria because they don't really have ways to support them in the gut. That's one hypothesis. So things like that are things that we're interested in, how these dietary substrates have these capacity to change our estrogen hormonal signalings for good or for bad, depending on a person's goal and needs, right?
Yeah, and so fascinating. that aspect and the tie-in with hormones, the tie-in with hormones and immunology back again also, and the microbiome. I know that there have been classic research where they've tried to either castrate males and show that their microbiomes become more female-like, and these types of studies where we're starting to tease apart sex-based differences in even our own physiology and our microbiome. Find out that females are not just males that have that one thing that happens once a month. Call the pharmaceutical companies and tell them that we need to be more inclusive of these in clinical trials and like these things. But what you are doing is so fascinating because you're really teasing apart this connection with hormones and how sex, gender differences impact the microbiome. I wonder if you could take us there and tell us a little bit about some of that work you're doing.
Yeah, for sure. Those are things that we're thinking a lot about nowadays. So my earlier work, we are interested in the metabolic phenotype, and in particular, that loss of ovarian hormones. So we overactimized mice using that as a model for postmenopausal women and looking to see, similar to the castration that you're talking about with that huge loss of gonads-derived hormones or sex steroids, what that does to the microbiome. Right now, we're really interested in to then connect it back. So the changes that happens that we observe from hormonal changes of the host and impact microbial communities, does that microbial community then further affect the host? So that's where this model that we talked about, the germ-free mice or gnotobiotic mice that we talked about, came in handy, that we've been doing studies looking at both male and females. When we overactimize, remove the ovaries of female mice, and then when we add estrogen back, so looking at that loss and gain of function, same in males, we castrated them and then add the testosterone back, loss and gain of function again, and harvest their gut microbiome. And then we then take those microbiome, inoculated them into germ-free mice of the same sex, but they are intact. They don't have any loss of their organs or production of hormones. What does that do to them in terms of their physiology and their metabolism? This was actually an unexpected finding that I had as a postdoc, where I did similar studies and inoculation, and I found out that one of the groups, their uterus looked huge when I dissect them at the end of the study compared to the other group. I'm like, well, I don't really know what this means. This has to mean something. So I quickly harvest the tissue and then collected data trying to understand, especially since the data that we saw were kind of opposite from what the donor's phenotype would be. So it would suggest that potentially the microbes is gathering information from the host and able to then affect the host differently.
Yeah.
So I actually took that data and went to find a reproductive biologist in Wisconsin and knock on her door. I'm like, I don't understand why I'm seeing opposite results. Help me out. And she think that it could be a brain and gonads connection that the microbes is able to intervene. So we think about gut-brain axis a lot when we think about the HPA axis. That's the more work done in depression and anxiety. This is what we call the HPG axis. So the hypothalamic, pituitary, and gonadal axis. So where our brain can tell our gonads, so ovaries or testicles, to make more or less sex hormones depending on the state that we're in, the needs of the hormones that we need at that time. So it seems to us that the microbes of a host where this HPG axis is disturbed, so that happened in many different phases in life, both in male and female, right? In puberty, that's where we started to have more production of these sex hormones. In reproductive age of women, pregnancy, huge changes in hormonal profile. Menopause, again, loss of that hormonal ovarian hormone productions. So a lot of fluctuations happen normally in our life. In males, throughout, and we don't have that, a significant fluctuation, but over time, your testosterone productions will go down as well. So what does that do to us, to the microbes, and then impacting us? So we're very interested in understanding this gut, gonads, and brain interactions. It seems like from the data that we acquire so far, it's true that, or hopefully it's true that the microbes is able to talk with our brain and impact that connections in ways that potentially, I'm hoping that we can then discover ways that we can manipulate the microbiome through either diet, nutrition, or even pharmacological intervention to then impact the microbiome and then impact our sex hormone profile, homeostasis, and that has many implications on various diseases, right? Not just the ones that have sex differences, sexual dimorphism. Most diseases actually have sexual dimorphism, but also reproductive activities. There's a lot more people having trouble with reproductions nowadays, it seems like, from back in the days. So is that something that's environmental? Is that something that we can manipulate by changing our microbiome? Maybe there's certain things that we should be eating more in our diet that can promote reproduction if that's something that people are having issue with. Or maybe the other way. Can we have a natural way to then suppress reproduction if that's what we want? So some of the things, those are some of the things that we're thinking about and trying to understand more.
That is awesome. That is awesome. And I want to keep asking you so much about the science because it's so great and it's so fascinating. But I want to also hear, like, what are the types of students that you get? Are these nutritionists? Are they animal scientists? Are they both a little bit like you're doing it all? And then bioinformatics and microbiome analysis and all of this stuff too, and collecting fecal samples or transplanting. I mean, tell us a little bit about what are the types of students that are attracted to work with you?
Yeah, it seems like we do a little bit of everything, huh? Jack of all trades.
I love it.
I feel yeah. The students that are mainly are in my lab are very interested in the nutritional aspect of things. How do we use nutrition to modulate the microbiome and impact the host? One of my students got very interested in the hormone aspect. So along the way, we're doing all of those studies, and she's very interested in endocrinology now. So I would say a little bit of all. One of the students that I just graduated, she came as a microbiology background from undergrad. So then her study is she's more interested in manipulating the strain of bacteria. So then we were creating different synthetic communities that has or does not have that equal conversion activities as a model, so that we can then test to see if that particular compound can then drive disease susceptibility or not. One thing that I forgot to mention is that equal conversion capacity that we mentioned, mice are thought to, at least the mice that we use in the lab, are thought to be very efficient equal producers, all of them. So the microbes that they harvest and naturally are all very efficient equal producers. So that makes mouse model a little tricky when we wanted to see if equal is the reason why we are or are not obtaining benefits from these dietary intervention. So by manipulating the microbiome, we can then create models that we know that do not have this capacity to make equal, or they do. So some of the things that we are able to do would attract a more microbiologist minded student as well on top of the nutritional student. And because of that sex hormone connections, and we're really at the early phase of understanding how microbes influence sex hormone homeostasis, that goes to more like biology. Pulse student might be more interested in that type of work, a little more basic than applying that to, you know, nutritional intervention later on.
But I mean, your work, even like this whole gut brain axis and the whole behaviors, and oh my gosh, like eating behaviors, sexual behaviors, or tendencies, or fertility, or, you know, maintenance of pregnancy, and nutrition of your fetus as it's growing, and right, and the hormonal aspect to maintain a healthy endometrium and a healthy, you know, pregnancy. All like, there is so much, and the preparing for the pregnancy, as you said, like maybe you need to be thinking about your diet a few months before you want to have a baby, and then you should be thinking about shifting to this diet once you have your, so like, this is like, it's incredible how broad it is. And so do you work with do you collaborate with clinicians? What are your typical collaborators there?
Yeah. So right now, even though that most of my earlier work are focusing on metabolic diseases, cardiometabolic impact, right now we're taking this question in a more approach that we're trying to understand how the microbes impact us, impact the host. So microbe and mammalian cells interactions on sex hormones. And then we picked, we look at the disease that would make sense in terms of that interactions that we discover. For example, one of the projects that I am collaborating with Doug Brubaker at the Biomedical Engineering. So he is a system biologist. He does a lot of informatic work, modeling. I had a random wild thoughts that I think maybe, so IBD or ulcerative colitis is a disease with sex differences. A lot of the autoimmune based diseases seems to be have a sex difference. It seems like from some of the data that are published out there, this is a disease that could be dependent on an estrogen receptor beta signaling and pathway, which is the isoform that are less known, but it's the dominant isoform in our gut. So I thought that it has to be something with the microbes, maybe certain microbial metabolites is able to bind to these receptors and affect disease susceptibilities. So we're doing some work looking at microbes, whether either metabolites or certain parts, whatever the microbe is doing is able to affect signaling. Yeah. And he's approaching it from a different angle, taking existing data sets of humans that have colitis or not, male and female, and with all the microbiome data, metabolite data, he's able to then try to tease out certain microbial metabolites and microbial taxa that are associated with disease or not, and also those that are sex dependent. So those that seems to be affecting males, not female, or the other way around. And then we can then take that into experimental setting in our hands to test to see if it's true in vitro, in vivo, that connection. So I work with people like that a lot that are very savvy in informatics, multi-omic analysis, and then how do we then test to see if that link is true? If that is true, then how do we use dietary components to impact that? And maybe that's a way that we can affect disease prognosis or prevention of disease, or are those targets that we can then pharmaceutically either isolate or create something that can then intervene with that connection in ways that would benefit our health. So I definitely collaborate with people like that. I collaborate with clinicians when it makes sense, right? These are things that we wanted to bring all the way to translational science. So I work with some gastroenterologists from IU School of Medicine, where they bank samples from IBD patients that we can then harvest microbial community from these people and then colonize mice and see if that community affect disease differently. I also, of course, work with people who are more basics, looking at metabolites, looking at gene functions, and more bench work, creating different type of community from single strains of bacteria type of work as well. So again, a little bit of everything.
Yeah, so good, so awesome. And you know, what an incredible opportunity or advantage to have all these engineers at Purdue that can help you, you know, with the machine learning, with the informatics piece and the modeling and all of that. I mean, and then also having the clinicians at IU School of Medicine right here and being able to work with them so closely. What about companies? Like what companies do you think would benefit from this type of work? Is it mainly, and I'm so naive in this area, are there food companies that are interested in this type of work? Is it probiotic companies? Who's interested in this type of work?
Yeah, I have some experience with some probiotic company that I'm working with that are interested in work like this, more like, especially where I'm at, our department has the capacity of doing control feeding or clinical trials where we have metabolic kitchen, we can design diets, so we can dose people a lot more precisely. We make their food. Those people, yeah.
With food.
with food, yes,
Yes.
more precisely. So we have companies who are interested in looking at how the probiotics can then support or being able to support certain functions. And that could be related to, don't really know how much I can disclose, but that could be related to certain disorders or issues that people might have encountering different type of foods. Can we design or identify bacteria that can then support that type of disorder and, you know, make the symptoms go away? Food company definitely would potentially be interested in, again, our capability, especially able to do control feeding trial, but also in mice, right? We're able to test these hypotheses in a more precise way, as well, by controlling the microbiome, controlling the diet, controlling the environment that they're living in. So we're able to tease out what exactly it is in that microbe and host interactions that the food or nutrition is able to affect and support. Pharmaceutical company potentially can be interested in, especially our hormone work. The ER beta work that I mentioned with IBD earlier, that's actually a target that a lot of the pharmaceutical companies are interested in identify agonists or antagonists that can affect that signaling pathways. There are a few other different pathways that we're thinking about that could also intervene with a disease prognosis as well. That might be something that a pharmaceutical company could be interested in as well. And that just, sex hormone itself has such a profound impact on everything. So I would think that that's something that one really should be thinking about. Not a lot of people are, for good reason. This is a very difficult things to manipulate, right? Because we can't remove it completely for a long period of time without consequences. So it's an interesting and fun field. I'm actually collaborating someone in Wisconsin doing similar manipulation in human, and we're trying to see if we're seeing similar changes in the microbiome from these human, and if we inoculate them into mice, we can see that gut-brain gonads interactions as well. But lots of things that
so awesome, so awesome. And you know, and as I recall the conversation, I mean, we went everywhere here, and I I wish we had a lot more time so that we could take of you to tell us more, because there's so much stuff. But I mean, you took us all the way in women's health. You took us all the way from, young stages of maturity to pregnancy and menopause, and you took us. It's such a wonderful journey to help us understand a little bit more, what is this technology, how much we are at its infancy stages of development yet. And thank you, Dr. Tzu-Wen Cross, Assistant Professor of the Department of Nutrition Science at Purdue University. It's been a pleasure talking to you.
Same. Thank you so much for having me. This has been fun.
Yeah, absolutely. And I hope you have a good rest of your day.
Thank you. You too.
Thank you.